Cancer is a class of diseases, meaning that hundreds of different cancers exist. There is no conspiracy from Big Pharma that they are hiding the cures that they can profit off of treatment. Some cancers are easier to treat and others are not. Cancer is a self-disease that occurs due to a mutation in a cell that somehow bypasses the internal checkpoints mentioned before. Oftentimes, the body's immune system can detect cancerous cells and destroy them before those cells replicate. But age, genetics, and certain environmental conditions, or carcinogens, things that cause cancer, can trigger a cancer mutation that can avoid the immune system. Once a cancer cell is able to divide, it usually does not stop resulting in tumors, and if left unchecked, cells of these tumors will metastasize or spread throughout the body, eventually leading to a person's death. So where does cancer originate? All of us have positive cell cycle regulators called proto-oncogenes, with p53 genes being the most well-studied. These genes dictate when cells divide and when they don't. In normal conditions, p53 genes detect when the cell is abnormal and has faulty DNA. It can stop cell division from occurring and either trigger apoptosis, or programmed cell death. That's right. Each cell has a self-destruct code to help prevent the spread of viruses and cancer. Or, the p53 gene will simply tell the cell to stop replicating and repair themselves before being allowed to continue with division. Now, remember that not all mutations are bad. Mutations are simply changes in DNA. However, when a proto-oncogene mutates into an oncogene, or cancer gene, the regulatory processes of the proto-oncogene are diminished. Mutated p53 genes are the most common oncogenes in human tumor cells. In healthy p53 genes, abnormal cells can be ordered to repair faulty DNA or be marked for apoptosis. Mutated p53 cannot order apoptosis or pause the replication to fix the problems of the cells. Thus, the cell cycle continues, which could lead to cancer. Now, while proto-oncogenes promote cell division in a normal sense, tumor suppressor genes are those that code for negative regulator proteins. In other words, they prevent the cell from undergoing division. And these are important when cells must be put in the G0 state due to lack of resources or faulty DNA. If tumor suppressor genes become mutated, its ability to stop faulty cells from undergoing the cell cycle is less influential, and thus cancer could ensue in the formation of a tumor. So now that we know the genetic causes of cancer, how is it that you as an individual get cancer? Well, many cancers are pretty much just brought on by who you're related to. If your parents or grandparents have cancer, your chances also increase, unfortunately. Environmental causes of cancer are called carcinogens, and these include smoking, asbestos, poor air quality, certain chemical exposure, UV radiation, and many more. Pretty much anything in the state of California causes cancer, as stated by their warning labels. All of these cause mutations in the proto-oncogene and tumor suppressor genes, which bring about cancer. Cancer starts off with a cell that is mutated to the point that none of the regulatory molecules are functioning. If it survives cellular division and is missed by the immune system, it will rapidly divide to form a tumor. Tumors require nutrients and gas exchange, and so growth factors are sent to the blood vessels to help feed this tumor. This is called angiogenesis. As cancer progresses, the cancer cells can then enter the bloodstream and spread or metastasize throughout the body. This is when cancer becomes difficult to control. The success of cancer treatments depend on the following. How far along is it? If you spot it early enough, you can prevent metastasis and go into remission, meaning that cancer is under control. Curing cancer is impossible because it is a self-genetic disease. You can only suppress it. How close to the surface of the body is it? Skin cancer is much easier to treat than pancreatic cancer, since skin cancer is easier to spot and pancreatic cancer is deep in the body, requiring MRAs or other technology. Also, age. The older you get, the harder cancer is to control. The more your cells divide throughout your life, the higher the likelihood your cells will produce defective daughter cells. And finally, the state of your immune system. Treatments unfortunately weaken all cells, including your immune system, so it makes it more prone to contracting other illnesses. Do yourself a favor and be sure to get cancer screenings and do monthly checkups in specific areas depending on your age.
Finally, how do prokaryotic cells divide? So bacteria do not undergo mitosis due to having no nucleus. The nucleoid, which is shown in the red circle, copies itself and distributes itself to the opposite sides of the bacterium. It then develops a septum to divide the cells, allowing the cells to break off of one another. So that septum is a portion of the cell wall. Some single-celled eukaryotic organisms also undergo binary fission as a means to asexually reproduce. While this and cellular mitosis can lead to making more cells, all the cells are identical to each other.
However, the key to life is genetic diversity. More diversity of genes amongst the population increases the chances of species to survive changing environmental conditions. But how do living things achieve that? In our next set of videos, we will learn the importance of sexual reproduction and how sex cells form.